I'm pleased to introduce our next speaker, Professor Simon Foote, who was elected to the fellowship in 2016, but wasn't able to attend that last year's gathering, and I'm pleased that he's here to join us now. Simon is interested in the genetic control of disease, with a particular focus on infectious disease. A major research focus of his laboratory is identifying new drugs to combat malaria. Thank you.
So thank you very much. Apologise for not being here last year. I also thank those who elected me last year. I walk past this building twice a day to get to the John Curtin School from home. And I must say that over the last 12 months, I've done so with a certain degree of pride as I walk past this building. So what I'm going to talk about today is one example of the work that my group is doing. It is essentially an anecdote out of a career working in malaria and host resistance genetics. And in fact, it's a topic which has very little genetics in it at all and was purely based on an accident. And I'll describe what that accident is shortly. But first of all, I am actually an itinerant researcher. I have wandered around the world, and these are some of the places here that I've worked, from Massachusetts at the Whitehead Institute, to the Weehai in Melbourne, to the Menzies Research Institute in Tasmania, Macquarie Institute, and I'm now at ANU. And I've enjoyed working in all of these places, and I've been extremely privileged to work with a large number of very talented individuals, and as has been iterated before, it is only through working in these large groups of very keen and often very young people that we've been able to actually do the research that some of which I'll report today. So one of my major interests is in malaria. Malaria is transmitted by the female Anopheles mosquito who bites you, as you can see here. That'll be an experience that all of us have shared. Fortunately, not many of us will share the next part of the story, which is where the mosquito transmits malarial parasites into the skin, which then migrate through the lymphatics to the liver, amplify within hepatic cells, and then burst, and then infect red blood cells. And the rest of the life cycle is one of reinvading red cells, multiplying and bursting those red cells. And it's that part of the life cycle that malaria gives rise to all its symptoms and indeed all its mortality. It also develops into a separate form, which is a sexual form, which is taken up again by the mosquito, and one sees a sexual life cycle happening within the mosquito. So we are interested in how the host actually combats malaria. And we've done this from a genetic point of view, but this story is actually about the humble platelet. Now, the platelet actually is normally used by the body to stop you bleeding to death when you puncture your vascular system. But, and this is a picture here of a platelet actually binding to an infected red blood cell. So we've actually found that there is actually a direct interaction between an activated platelet and a red blood cell carrying a malarial parasite. There is a lot of literature about parasites and malaria, and there's a relationship between the level of parasites in an individual and an inverse relationship and the number of platelets. So the more parasites, the fewer platelets. And if you look at an individual, this on the right here, where you actually see a decrease in the number of platelets, i.e. thrombocytopenia, as the number of parasites increases. So there is actually a relationship between the two, and it's been known for quite some time. And we've been able to recapitulate this in a mouse model of malaria, where you actually see, and as the parasites increase, you actually see a concomitant decrease in platelet levels. Now, this is where the coincidental accident occurred. TPO is a molecule that regulates the production of platelets, and it does so by stimulating platelet production from the progenitor megakaryocytes, but it also produces more megakaryocytes as required, megakaryocytes being the large cell that produces platelets. So Warren Alexander at the Walter Neiswell Institute was working on TPO and knocked out its receptor, which is actually C-MIPL, And when you do that, you end up with a mouse that has 10% the normal number of platelets. Wrong way. So we accidentally infected these mice. So they look like normal mice, except they've got Mipple gone, and the mouse house screwed up and gave us Warren's mice instead of our own mice. We infected them with malaria, and lo and behold, they died, when in fact they should have survived. So this is an example where you can actually see that these survival curves, you end up with a significant death rate in mice with low platelets. To inhibit the action of platelets, one uses a number of drugs, but aspirin is a classic, right? So you can take aspirin if you've got evidence for heart disease, and it essentially inhibits platelets, which decreases the thrombus production, which is also a side effect of platelet activation. So if you give mice aspirin, then they die more. They're more susceptible. And that's because you're actually inhibiting the action of platelets. Platelets can no longer protect against malaria, and therefore the animal dies. We can see this in humans as well, either in vitro or in vivo. You give humans just a normal dose of platelets, of aspirin, the sort of dose that you would actually take if you were taking it to prevent thrombus formation. And you take that blood out and you try and infect it with malaria. And what actually happens is that those platelets can no longer inhibit the growth of malarial parasites. So platelets are obviously very key in actually stopping malarial parasites growing in culture and also in our mouse models in vivo. So the question that then becomes is, how is this happening? What is the molecular basis? How do platelets actually inhibit the growth of malarial parasites? So this is essentially a summary slide of what's actually happening, and one can see two lots of platelets. So there's some platelets in the corner over here that are actually binding to an uninfected red blood cell, and they have not been activated. So you can actually see that all you can see there is a platelet molecule called platelet factor 4, and it is retained within the platelet, and it's not spread anywhere. On the other hand, in the centre of the field, you can actually see, again, a platelet binding to a red cell, but this time this red cell is infected with a malarial parasite, and we've stained the DNA of that parasite blue, and you can see that the platelet factor 4 is now spread around the red blood cell because the platelet has been activated and has actually released the granules within the platelet which contain this molecule, platelet factor 4, and that has actually spread around the infected red blood cell. So platelets actually are intelligent. They can differentiate an infected red blood cell from an uninfected red blood cell when they bind it. And we can go on and show that that parasite within that PF4 cell is actually dead. So this activity of the platelet binding, activating, kills the actually parasite within that red blood cell. And we've also gone on to show that the molecule that does the killing is in fact platelet factor 4. So platelet factor 4 itself will actually kill parasites within a red blood cell. Platelet factor 4 is what we call a cytokine. And these have a very wide range of effects. There is actually a famous receptor on the red blood cell, on many other cells, called Duffy, that actually binds cytokines. And Duffy on the red blood cell actually binds platelet factor 4. And we've demonstrated, and you can see here, that platelet factor 4 gets into the red blood cell through this receptor called Duffy. Because if you take a normal red blood cell that actually has Duffy on the surface, then you can actually see inhibition of growth and killing of the parasite. Whereas if you look at a red blood cell that genetically lacks Duffy on the surface, then platelet factor 4 can no longer kill the parasites within those cells. So Duffy is obviously important in bringing it in. So this is essentially our model. Platelet binds, activates, produces platelet factor 4, which binds to Duffy, gets into the platelet factor 4 gets into the red blood cell through Duffy and kills the parasite. Haven't got time to tell you how it kills the parasite, but I've also got a Simon Hay slide that Jane actually used as well to show an interesting coincidental fact. Duffy negativity, i.e. the lack of Duffy on the surface of red blood cells, is essentially common. In fact, it's more than common. All of sub-Saharan Africa is actually, that red bit up there, is actually Duffy negative. There's virtually no people living in that area that actually have Duffy on their red blood cells. That is also the major site of malarial mortality and incidence in the world. And there appears to be a much greater incidence of death given the frequency of infection in Africa than in any other part of the world, including Papua New Guinea, which is near us. So there's an interesting story to be actually worked out here. Is the incidence of malaria in Africa due to the presence of the Duffy negative allele in Africans? That is my story. And these are a number of people that have actually worked on that particular project throughout the last 15 years. So thank you very much for listening.
We have time for questions. If I can. Yes, there.
Sorry, why do you think...
Can you wait for the microphone, please?
He's got a loud voice.
Nick Martin, Brisbane. Why do you think there haven't been mutations that have been selected for Duffy positivity in sub-Saharan Africa?
So we don't know, and that's a big question. So the putative selection for Duffy negativity is Plasmodium vivax, which actually gets into the red blood cell via Duffy, so that's its portal of entry. And there's no evidence, but there's a theory that Vivax, which is not particularly lethal, was actually more lethal at one stage. So it's not obvious that Vivax is still producing an effect. We don't know. It's possible that those mutations haven't actually gone in just through lack of migration over the last 10,000, 20,000 years. We don't know.
We can take one more question, if I can see. Yeah.
There are so many things that inhibit platelet formation and dampen it down and so on. In this is one case where you really want it, I take it.
Yes.
So are you following on to look for ways of encouraging platelet formation, production, I should say, without the side effects that you'd otherwise get on clotting and so on?
Yes, well, just incidentally, the treatment for malaria in most countries with a low health budget is aspirin to decrease the fever plus an antimalarial. So our work would suggest that aspirin is probably not a good drug to give someone that's actually got malaria. And there has been a shift in using paracetamol instead of aspirin, because paracetamol doesn't do this.
Okay, thank you very much, Simon.